happy to have Dr. McClure presenting for us from the USA Dairy Forage Research Center in Madison. I'll let her introduce herself and get us started with the seminar. Please, Dr. McClure, the floor is yours.
Thank you so much. I very appreciate being here. I always have to start all of my presentations with this little disclaimer slide. So it's been up for a while, which is great. So here we go. Today I'm going to talk to you about genetic defect recording and the search for something nobody wants to talk about. So to give you an idea of what we're going to talk about, I'm going to cover my background, why we should search for defects, building a genetic defect database from scratch, case studies from the past, both my first and one of my very last cases, some historical data, and the hardest part that you tend to run into, which is getting farmer engagement. So in terms of background, my bachelor's is in animal sciences at the University of Missouri. I followed up with a PhD in veterinary parasitology, where I worked on parasitology, microbiology, infectious diseases. I then moved on to Baltimore, Maryland on the East Coast, where I worked in a BSL-3 facility and working on both epidemiology and also tick immunobiology. And while I was super excited to work in a BSL-3 environment, I thought that was like the coolest of cool things. After you sneeze in a PAPR, it becomes way less cool because you end up having to like run out and disrobe so that you can clean the front of your mask off. So I was a little disenchanted by that. But following that, I got the opportunity to move to Ireland. My husband is also a PhD. He does cattle genetics, and they were looking for a geneticist. And when we went over there, they said, hey, we're interested in health and disease. Do you think you could help out with that? And so I said, yeah, of course I can. So I started working with them and trying to get essentially working off the stuff they already had to collect health and disease traits from farmers, turn that into genetic evaluations based off of those recorded traits, and then talking to them about some of these genetic defects that came up. I thought, we really need a good way to record this and to get farmers on board with it. So I worked on that, and then a little bit of public policy and a whole lot of knowledge transfer, because communication is extremely important in agriculture. So where I'm now is at the USA Dairy Forage Research Center. I put a little map down here, which shows if I were to drive from where I'm at to where you are right now, it'd take about 9.5 hours, which is a very long car ride, but doable. I'm glad I didn't have to make it for the talk today. But some of my select research projects, everything focuses around the rumen microbiome and the efficiency and making cows more efficient, feed efficiency, and also health, because the microbiome impacts that a lot. I'm a big fan of methodology work because as a public researcher, I'm given the chance to spend time on things that may not be the sexiest research, but it's critical for getting the best research. So I spend a lot of time working on methodologies. But I'm also looking at assembly of metagenomic assembly of rumen microbes, the role of protozoa, some non-invasive methods for rumen sampling, because you can't just put cannulas in every cow you see. Farmers will frown on that. And also whether or not that process actually changes the rumen microbiome, because we're going from an anaerobic environment and adding a hole in it, which is going to add a whole lot of air to it. And then way further down is genetic defects, because this is just a passion of mine. So I'm very slowly kind of dipping my toes into this in the United States. US is way bigger than Ireland. So this is a massive project. So why am I interested in searching for these genetic defects? Well, one of the best examples is this article that came out in The Atlantic in 2016 talking about Pawnee Farm Arlinda Chief. Some of you may have heard of Holstein haplotype 1. This guy is the progenitor of it. And this article sensationalized the fact that he cost the dairy industry $420 million from a single mutation in this bull. That sounds like a lot, and it is a lot. But if you actually read the article, they end up saying that, hey, despite this lethal mutation, they ended up adding 30 billion to increase milk production over the past 35 years. So while he cost 42 million, he gave 30 billion to the industry. So what does that tell me? It tells me that we need to manage these, we can't necessarily get rid of them. And I'll talk about why that is in a little bit. But if we manage these genetic defects, then we can end up getting those genetic gains without the losses. So 2015, 2016, this is 10-year-old data. So I've been away from Ireland for a little bit. So this is old data. But back then we calculated that Ireland was losing 1.7 million euros a year to these genetic defects. And in the United States, they were losing about $11 million every year for genetic defects. So after all this, and we started implementing breeding schemes where you could match up sires and dams that do not have the same genetic defects, that actually has gone down to 4.1 million in the US. So as we're finding these, as we're able to put them on a SNP chip or have individual tests for sires and dams, we're able to actually decrease this. Now, why is it not zero? Because new defects pop up all the time. So in that, all animals carry a number of lethal recessives. And the point here is recessives. So it's not going to show up unless you accidentally mate two of those carriers together. It's estimated that 1 in 2 humans carry a lethal recessive as well. Many of the animals are carriers for different disease alleles. So again, management is key. If you're not breeding animals that carry the same ones, you're doing pretty okay. In 2024, Cole et al. Put out this great paper where they were talking about how new alleles are found pretty much so every year. And you can see from the graph that we had a pretty big spike in around 2014, 15, and then it's kind of dropped off since there. That doesn't mean that they're not out there. It just means that people aren't doing as much research on it. So they are there. Every year they're being found. We may just not have the cause of mutations yet, therefore no paper on it. The other thing that we have to keep in mind, as inbreeding coefficients increase, in theory, more defects will pop up because then you have a greater chance of mating two carriers of these recessive alleles together. So what's with inbreeding, right? Why are we doing this? Sometimes there's intentional inbreeding, and we call that line breeding. I put up a quarter horse pedigree here. And anytime you see kind of a color on the end, that is an animal that's in the pedigree more than once. So you can see it's pretty colorful here and here. As we get closer up to the actual animal itself, it's a little less, but we have a very strong foundation of inbreeding. So with that in mind, these will be fairly inbred. But not everybody's doing that. Maybe you have a rare breed with limited populations. I'm thinking about in Ireland, the Kerry cattle. There's not a ton of them out there. So you only have so many animals that you can breed together to maintain that pure line. The other option is starting to breed in other animals that may be similar to expand that genetic population. And finally, you can't avoid what you don't know. Sometimes an animal will come to you and they'll have an incomplete pedigree, or worse yet, pedigree error that you may not know about. So sometimes this stuff happens unintentionally, or if you don't genotype the animals, you don't know the status of one of them when you breed to the other. So bottom line, why are we searching for these genetic defects? Farmer losses and the economic impact, as mentioned with the HH1. Loss of genetic gain. So this is a picture of Angus cattle through the ages. These are the top bulls from essentially 1938 to 2025. And you can see that conformationally, they look very different, which I think is really cool as well, because this was just done with natural mating. But with the advent of gene editing, if you have a superior sire, you could actually edit the gene out that is the bad gene or the lethal recessive, and then you can change your population. Now, we have some issues with that as well because of regulations and stuff, and whether it's genetic modification, but it is possible to do something like that. Lastly, one person's defect is another person's pet. So many people are really interested in these adorable mini Highland cows, and often they will carry a dwarfism gene. And so people are specifically trying to breed the dwarfism gene in there. As a responsible breeder, I don't recommend that because that dwarfism gene ends up causing health problems later on in the animals, and you could have other problems. But this is a very expensive fad at the moment. Before I looked into it, getting a mini Highland cow was going to cost me like $6,500, and there are many more things I'd rather spend my money on. But people will pay that for one of these silly little cows that's just a pet. So how did the actual genetic defect recording system happen? I had it on my mind for a while when I came over to ICBF because they have such a great database of all sorts of things that they're bringing in. And so in 2013, I attended the CAVI conference, which is a Veterinary Ireland meeting. And I talked to the vets and I said, hey, I'm interested in doing this. You all are the boots on the ground. I'm really interested in talking to you about what you've seen, what you're interested in. Have you seen more of one thing versus the other? And I ended my talk with this slide. Yes, that is me sighting in my deer hunting rifle because I am disease hunting. We are no longer hunting deer, we are hunting diseases. So we are actively looking for animals with the following conditions. And so I had this list of conditions that I had seen on farms or ones that we had SNPs or causative mutations figured out for, but we hadn't validated them yet. So I needed a population to validate those SNPs on. So I said, hey, farmers, talk to your farmers. If they send you out and say, hey, they've got something funny, please send it on to me. Let me know. And so that was in October 2013. A month later, I got my first call. So I was contacted by this farmer. He says, can you help me? I have a problem with my cattle. And his vet, he called the regional veterinary lab vet out, and he diagnosed the animal with progressive ataxia, which I was like, yeah, that is known to be a genetic defect. We just don't know a whole lot about it. And at this point, I'm going to pause you on that because that one call lit a fire under me so that I had to get finished with the survey and make this available to all the farmers. So I created a head to tail survey. I started out with a survey that was from the French regional vet labs that they would mail out to farmers and have them kind of list what was going on with their animals. And it was often technical terms. So I don't even know if it was sent to the farmers. It may have been sent to the vets. And the vets would actually fill it out instead of a farmer. So I took that, a lot of Google Translate, because French, as much as I think it's a beautiful language, I am not good at it. Even my Spanish is not great. The United States is not known for being very good at speaking multiple languages. Sometimes I even struggle with English. But we turned this into a collaborative effort. So I spoke to farmers, I spoke to vets, I spoke to the regional vet lab who were kind enough to send me a giant list of defects that they had come across over the years. And from there, I made this particular survey that started out asking a number of questions and then moved on to talking to farmers and asking, like, pinpointing more about it. So for this, one of the main things that I did is I turned it into common vernacular. One of the diseases I was looking for is atrasia. This can manifest to what the farmers call water belly or no back passage. And what it is a constriction somewhere between the esophagus and the anus in an animal. And so with that, the animal's born, seems totally fine. They take their first drink of milk and they can't pass anything. And so they end up dying. This is a big problem. I'd seen a number of cases like this across Ireland, and so I really was interested in that. So how do I do that? I use the phrases that the farmers use. I don't use the vet talk. So I talked to the farmers, I did some of the most common defects, put them on here, and then I started searching for patterns. So what were some of these questions that I was focusing on that weren't based on the animal? First of all, I asked if the person was the animal's owner, whoever was recording. Are they the animal's owner? This is super important because then it tells me, is it a vet or is it a farmer who's actually giving me this information? Followed by herd number. In the past, Ireland, their herd numbers, I could basically get down to kind of a townland level by looking at the herd number. Why is this important? Because if you have a cluster of defects, they may not be genetic. It may be something environmental. So knowing the area around and knowing where these were coming from was important. Did I need to know the specific farm? Not necessarily, but it was helpful. Then I wanted tag numbers for the sire, the dam, and the affected animals because I could then use that to find their pedigree or find any information in the ICBF database on them so I could start searching for a pattern within the genetics of the animals themselves. Then we asked single birth, multi-birth, sex of the animal, and age of animal. All of these are important because some defects affect different ages. And it, again, creates this picture, this full picture for me to be able to better understand what's going on. That is followed up by the head to tail. Do you see something wrong with the head? And then it goes into more depth, the eyes, the nose, the mouth, and it has the farmers click these boxes. And if they need to, they can fill open field text, but I want to warn you, if this is something you're interested in, open field text is both the greatest and the worst because nobody spells anything the same way. Everybody's got a different word for something. And so it's going to take more translation. Then finally, the last things that I asked are, can I have a picture of a video of the defective animal? Can I have a DNA sample? So send me a little snip of the ear or something like that, a small tube of blood, anything along those lines. Unfortunately, some farmers got a little overzealous and I did receive entire ears in the mail. After about the second time that happened, the front office stopped opening my mail and would just leave it on my desk because they were pretty grossed out by that. And then the last thing is, can I contact you if I need more information? The reason this is possible, or sorry, was important is because sometimes these defects were looked at as being shameful. I found that out. I didn't understand why, but they were kind of ashamed of the defects. And so sometimes a farmer just wanted to be like, here's my information. I don't want to talk about it, but do with this what you want. So I wanted to give them that option so they felt like they had some ownership over this as well and how much they wanted to participate. Now, with that being said, it was very rare that they said, no, don't contact me if I need more information. Most of the time, it was a yes. So it takes a lot of reports to identify a genetic defect because sometimes a defect isn't a genetic defect. So searching for these patterns, I'm looking at breeds, I'm looking at pedigree, and then I'm also looking at those external factors, location. If the farmer said, yes, I can contact them and I'm not seeing anything like it, sometimes I'll ask, hey, was your dam treated with anything during the pregnancy? Also, management. I can talk to them about their management styles. Hey, what was done with this? Was this cow treated any different than the rest of the herd that had this? It offers opportunities for you to dig deeper and to kind of be a detective about this. Now, the sad thing we found out is unless verified by genotype, pedigree might not be correct. In 2023, a paper came out saying that between 11 and 20% of pedigrees are not correct. And that is kind of an abysmal number. So this goes back to that you may not know what you're looking for because you may not have the right pedigree. And the bottom line is you may not get enough results for years. This is a slow burn. This is not something you can do very quickly and like get off the ground and immediately have results. That's going to be very rare unless you have like a couple farms that have just had a ton of these because they all use the same sire. So why did this work so well in Ireland? Well, all animals since 1998 have been tagged. So that means we've got great records on almost every single animal that's there. All movements are recorded, which is great because if I'm seeing a rash in a specific area, but one pops out in a different area, I can check to see if that animal came from the area where we saw all of the events, or I can see where that animal is born. Maybe it's something on the farm. For mandatory recording at birth, every farmer in 2015, mind you, had to record the dam tag, the sire, breed, calving ease, and the sex of the animal. That's a great start for anything. Optional recording was genotyping, sire tag, and so much more. So this allowed me to get this insanely good picture of what was going on those farms with those farmers if they chose to share. But there is always a downside to go with the upside. So the positive, you can get disease and outbreak tracking because of all of those animal movements. You can get traceability, which is amazing. In theory, if you have huge data sets that go along with every animal, it causes huge progress. But you also have to be smart enough and have enough wherewithal to dig through all of that data and wade through it. It's like a tidal wave of data. It's amazing, but it's also intimidating. And in theory, you should get reliable breeding values. But as I mentioned before, pedigrees aren't always correct. The negative, at the time, the department had a rule that if there were greater than 3 sets of twins born on a farm, they would essentially go visit the farm to see if there was something funny going on. So sometimes farmers would put in that the dam was something else. Back to the pedigree errors, see? There were specific restrictions on herd movement if they thought something was going wrong, which that's not really a negative because it can help out everyone. If, say, a disease outbreak occurs, it means that it's going to be contained better. There should be strict recording of farm information, medications, and birth, which is really, really helpful. But anytime there's a human involved in data recording, there's a possibility for it to be fudged or to keep them out of trouble, put together hastily, or it may not be accurate. And so one of the funniest examples of that I have of this is a paper from Japan came out and they said that more calves were born during a specific lunar phase. And I was like, dude, we've got all this data. We could check it out. And so my husband worked with one of our data analysts, Johnny Mack, and he looked at when calves were most often born. And what he came back with was it had nothing to do with the lunar phase. It had to do with the day of the week. More calves were registered on Mondays than any other day. So we kind of figured out after talking to a number of our friends who are farmers, I'm like, oh yeah, if a calf's born during the weekend, we'll just mark it down on Monday and it's fine. So then we send them all in at the same time. We don't have to worry about it. And then sometimes farmers would jokingly say that like an animal was born in the rushes and they couldn't find it. And so you'd end up with animals that were sometimes beef animals. This would happen. They'd be born and they'd be like, oh my gosh, when did you show up? Ah, sure, it's today. So again, anytime there's humans involved in any of this, that means that you're going to get a little bit of fudging. So digging deeper into finding these defects, basically what we want to do is we want to get an ear snip from the animals, extract DNA, sequence the animal or genotype the animal, depending on what we're looking for. You get an absolute mountain of data. You get a monster bit of data. We then take that data and compare it to the reference genome. Following the reference genome, we go back and forth looking for potential alleles that may be causing this. This can result in multiple SNPs. So then you hope you have more than one instance of this genetic defect so that you can then take it from the whole population and line those up and say, okay, which are in these, but which are not in the unaffected animals. And this goes back and forth a lot until you potentially find the causative SNP or SNPs, because it may be multi-allelic. And once you can identify what the causative mutation or mutations are, then it can be rolled into a test, or you can use it for SNP chips. So basically you're going from the farmer with the sample and back to the farmer with the test. And this is where like it gets really exciting. So while we were there, we actually helped develop the International Dairy and Beef SNP chip. So this was the IDB v3. It was developed by ICBF, Weatherbees, Chagas, and others. And we developed it with imputation in mind. So a few number of SNPs that could be imputed up to a higher density. And it had 72 validated disease SNPs and traits on it. At that time in 2015, 330,000 Irish and European cattle were tested annually with this chip. So that's a good number of animals getting tested. Weatherbees ended up commercializing the chip, which meant that it was available internationally for anyone who wanted to use it. And when we developed this, my husband and I actually wrote a little booklet so that we could share kind of all this knowledge so anyone could replicate these disease SNPs and traits. So this is the booklet I'm talking about. It is still available on the ICAR website. It was published in 2016, so we're 10 years on. We made two different editions. There's a farmer and a research edition. Basically, there's a small primer on genetics, a simplified version, followed by a blurb on each of the defects, the causative SNP, and citation. That's the researcher edition, which is here. The farmer edition left out those causative SNPs, and the blurb for the defect was using common language rather than vet speak. So essentially, this gave them a way to take a look at what this is. Often, if we could find a picture, we included a picture in there as well. And so farmers could reference this when they would get their data back. So the idea is to eventually update and republish this in the works. And it's in the works, but I will tell you it is very far down on my list of to do. So hopefully I will find and make time for that. Maybe that's what I'll do for a staycation. So very quickly, this is a list of what went on the SNP chip. We had lethal, we had unwanted, and then these are more the positive things for milk, color, beneficial like pink eye, resistance, and then meat. What I want to point out too, though, is anything that's in pink requires extra feed due to IP. And I'm really disappointed in this because I think that we shouldn't be patenting necessarily these ones. The ones that are specifically for making money, meat and milk, totally get that. That's a profitability thing. But I feel like really these lethal and unwanted ones, we should really have those available for anyone so that any animal can be checked without any additional fees because this is for the good of the farmer. So let's talk about my first case. That one that I was contacted in 2013 about. It was a farmer. He says, can you help me? My cows are falling down and I just need some help. The cattle were diagnosed with progressive ataxia by a regional vet lab vet. If you don't know what this is, it's essentially a disease that starts with a wobble in the hind end and it moves forward in the animal until they cannot rise and they will eventually die. It's a paralysis. And so he just wanted to know what can be done because he had bred these Charolais animals. It happens when they're 18 to 24 months. So this is right around the time they would be going to the abattoir. And he had spent all this money on these animals only to lose them at the very end. So luckily, I was given this video by John Fagan. Let me see if I can get this to, oh, I think I have to turn off my pointer, a laser pen, and see if, there we go. So this video, you can kind of see she's walking fairly normal, but the farmer's going to excite her. And this is where, as she's getting kicked by the other cow, you can see she's kind of wobbly in that hind end. And if I had the volume on, you'd hear the vet going, oh yeah, it's in her right hind end, it's in her right hind end. And so you can see that big wobble. So eventually this poor cow would end up laying down and dying. So the plan of attack was for the farmer to collect tissue from these affected animals. We're going to extract the DNA. I was doing exome sequencing because at the time we were kind of poor, couldn't afford full genome sequencing. And I had read a paper saying that 85% of the disease-causing mutations are found on the exome. So I was like, okay, if you're going to stack the cards, stack them in your favor. We ran it through a data pipeline, and we actually identified some potential disease-causing SNPs. So the next steps were to test and validate the correct gene and then roll this out to farmers. So during this time, we did actually identify 10 SNPs. And my husband took this research to a conference with the video and was showing it and being like, hey, we're looking for this, blah, blah, blah. And this group from France actually came up and they said, hey, we've been looking for this too because Charolais cattle, and we found the actual cause of mutation. So it was one of our 10, which is great. They published the paper and they were kind enough to let me send them DNA from these affected animals. With that, we were able to confirm that the sire, the dam, and half the sibs were carriers for this genetic defect. So that meant that this could get added to the next SNP chip so that we make sure that we're not breeding this into the Charolais population and racking up the amount of carriers that have this, because it's absolutely devastating for the farmer. So over 4 years, I had over 400 cases reported to me, which was phenomenal. It seemed like every year the number of cases that I got doubled, which is great. But after that 4 years, I moved to the States and it kind of languished for quite a while. But before that, and literally right before I was getting to move, I had a farmer call me and he essentially says, hey, I have these heifers, but they don't have udders. And I was like, okay, did they not like, are they not springing? What's going on? He goes, no, 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 no. They calve down just fine, but they have no udders. So I went from kind of looking like this to very much so looking like this, going, oh, they calved down? And he said, yeah, they calved down just fine, but they have no udders. I was like, oh, okay, this is really weird. So I said, all right, how many are we talking? He said 19 total. He had 10 from the stock bull from 2015 and 9 that he had bred the same stock bull to that came out from 2016. So well, what do the calves look like out of those, first calvers? And he said, they look pretty normal. Most of them were crossbreds, but, they're a little bit smaller. I said, all right, what about the rest of the cows? Are they normal otherwise? And he's like, well, I decided to get rid of the stock bull because some of them were smaller than I would have liked. And so I didn't think that he really fit in our breeding scheme. And I was like, all right, okay. So as I mapped it out, essentially, these are the 2015s. 3 of them had quote, quote, normal udders. And I'm putting it in quotes because the udders were smaller than what we would expect. And when I brought my vet friend out, she was like, these are a little hard feeling, almost like a mastitic udder, but with no heat and no actual mastitis. But they ended up having 2 fairly normal looking calves and a male. 2 of them he didn't even bother breeding because they were just small. And then the 4 that didn't have udders, they had 2 males and then they had 2 calves. And when we looked at those calves, they didn't have nipples. They had nothing. So this was really kind of crazy. So we did plan a farm visit. I asked a collaborator of mine who's a vet to come out with me. So she came out and we decided to take some samples. We took some pictures. We took blood to check for the various inflammation markers and also some toxins. We took tissue for genotyping and also parentage verification. We decided to genotype all the calves and the heifers and then look at When we looked at the blood counts and the toxin levels, everything came back normal. Nothing was wonky. So I'm going, oh, this is really weird. So we wanted to do whole genome sequencing on the affected cows and calves. So these are some of the pictures we took. Now I'm going to warn you, they're not the greatest because these were not super cooperative cows. So I had a selfie stick and a really old cell phone that I was just like shoving under them and hoping for the best. And it got mangled in the process. But all of these animals had calved down between 5 and 6 weeks prior. And as you can see, particularly 1644, there's nothing here. It's just fatty, loose skin. For this one, they had some like flat, possibly button vestigial teats. Maybe they were inverted. And then for this one, it was another flappy skin, perhaps one small nipple. So these animals had babies, but there was no udder there, making them the world's most useless dairy cows. Looking at the calves, I took a peek at the 2 crossbreds that came out, and there are, oh, I noticed that I'm not using my laser pointer again. Sorry about that. There are no udders here at all. There's no nipples, nothing on these 2 ladies. So I asked the farmer, can I flip one of your normal calves? Will it stress them out? He's like, no, go ahead. And this is what you would expect to see. You can see cute little nipples there. These guys had nothing at all, and they were female. So that was really crazy. But the farmer was excellent. He was very helpful every step of the way. And when he decided to send off those cows to the abattoir, he called me up and said, hey, these guys are going up to Donegal. If you want to meet them there, this is when I'm sending them. So I contacted the abattoir, and they were kind enough to outfit me and put me up in a little raised elevator. And as these cows came through the processing line, I just talked to one of their guys and I was like, can you just lob me off a chunk of this area? And so, very kindly, he just lobbed me off a chunk of where an udder should be. So I put them in my little baggie, zipped them up, labeled them really well, and then dropped them off at my friend's lab, which he was a regional vet lab, and sorry, collaborator, friend. And he essentially saw this for those 3 cows that I showed you earlier. What you can see is there is nothing going on here. This is just fat. There's absolutely no tissue development here. Luckily, another cow from the same herd went through, and this is what that looked like. So she was one of the unaffected ones. And as a control, this is what you would expect to see with udder development, not this. So this pretty much so said, okay, this is definitely real. This is something. We pretty much so decided this must have been a de novo mutation within that specific sire. Why did we think it was a de novo mutation? Well, the grandsire was pretty prolific. So we had lots of records from other stock bulls and also any female progeny that he had. And none of them had had any of these problems. So this is just literally a case of really bad luck. So this guy ended up with just one bad animal. It ended up costing him about $20,000 in replacements. Sorry, 20,000 euros. And that's because it was 2 years of replacements. It was his 2015 and his 2016 group. But none of these animals were normal. So he ended up getting rid of all of them. So at the time, we were looking for the gene of interest. We were trying to get it sorted as best as possible. And the thing that stuck with me the most was, what if this had been a stock bull? Like, can you imagine if this was an AI bull that carried this mutation? How many thousands of straws could have gone out and gone to many different farms, and the economic impact of that? So that's why this is so important. It's important to identify this so we can make sure that everything gets tested before it goes out. With a de novo mutation, it just unfortunately happens. But if we can catch those and find the cause of mutation, we can then make sure that this doesn't happen again. So looking at historical data from 2015, so these are the animals that were tested with our SNP chip, and these are specifically in AI sires that we had tested on the SNP chip. And what I want to point out is we had some decent rates of some of these genetic defects in a number of these sires. We're talking for Blad, 3.45%. For Jerseys, developmental duplication, 4.9, almost 5% in Angus. The Montbeliard one gives me a heart attack because nearly 30% of the Montbeliard sires that were AI were carrying the mutation for Montbeliard haplotype 2. So that gave me a bit of pause. When we went into the purebred population that had been tested, again, that Montbéliard haplotype 2 is super duper high because that carrier gene can be passed on from the AI sires. So this is just reinforcing that whole notion of get your population's genotype and check, breed smarter, do selective breeding. And then looking at a crossbred population, the commercial herd, because hey, crossbreds hybrid vigor. You still will see it within the herds. And that's because a bad allele can get passed on to a crossbred just as easily as it can be to a purebred. And so if you have things like Kiwi crosses where you're crossing Jerseys and Holsteins, you hope you get all the extra butter fat and that from the Jersey with all the extra milk from the Holstein, but you may also end up in getting getting the progeny having Holstein haplotype 3 or 1, 2, 3, or 4, and potentially also carrying Blad at the same time. So if you breed them back to a Holstein or a Jersey, then you're going to potentially see that genetic defect. So all of this is coming back to just being aware and training and to convince farmers just to breed smarter. So this is what I was kind of calling this. I was telling farmers we need to breed smarter. So if you have a high level of genotyping, it allows for this targeted breeding. You choose your sires and dams that don't carry the same genetic defects, and then you educate the farmers on how to do this and why to do this and to change the breeding strategy so that you can make sure you're not breeding carriers to carriers. This will then end up with more profitability for those farmers. The good news is computer algorithms and AI will help this because they can do that so much faster and pair up these matings so much faster than I can in my head. But we have to make them aware that every year new genetic defects are being discovered. So if this happens, you may have to periodically either regenotype an animal or send off for a specific test if you are concerned about it, particularly if you're a purebred population. So the last bit of this is devoted to the hardest part that I ran into when developing the system, and that's getting buy-in. As I mentioned before, genetic defects can be looked at as shameful. Like farmers really didn't want to talk about this. They felt like it was a failing on their part, which again, I really didn't understand, but you can't change how people feel sometimes. So you just have to be understanding for that and help them kind of overcome it and tell them, this is going to help in the long run. So I'm going to give you Jen's 7-point plan to encourage participation. So number one, make yourself visible and reach out to everyone. I had a ton of popular press articles come out. This is just one of them. And so it got the word out. Hey, this person is looking for genetic defects. This is important. We're going to help farmers. That's the bottom line. I did radio interviews. I went and I talked to vets. I went to vet events. I made sure that I talked to the people who were seeing this. I went to farmer events. I went to the plowing championship and I was talking to anyone that would spend 5 minutes with me about what this is. I went to young farmer events and talked to the younger farmers. So they have I think it's just called the Young Farmers. We call FFA here, but they're the next generation. So maybe they can help get rid of the stigma of shame associated with genetic defects. So talk to them, explain what's going on, talk to them about how cool this really is. I engaged with Chagas, which is kind of their branch of research for the country. Agriculture researcher, I'm with USDA, that's ours. They have Chagas. I engaged with the Department of Agriculture directly, talking to the regional vet labs. I engaged with breed associations, and I will tell you, they were some of the hardest to sway. And the reason why is because they felt like I was picking on them and saying, hey, your breed has these problems. But what I try to explain to them is, yes, your breed has these problems, but if you hide it, and then everyone who breeds these animals finds out that you hid it, and they're taking a large economic hit because you chose to hide this, that means that they are not going to trust you and it's going to get really ugly. I think it's also important to engage with AI companies or nuclear herds. That's what we call kind of the breeding herds here. Ireland uses a lot of stock bulls. So engaging with the people who are making those stock bulls was super important. Number two, use common, simple language. This is a fine line. Too simple, people feel like you're talking down to them. Not simple enough, it'll go over their heads and they'll stop listening. I'm hoping I have not done that to anybody in this talk, but I also don't want anyone to ever feel like I'm talking to them like they're stupid. Human nature is to be afraid of the unknown. And I am coming at them with something that is totally new, totally different. And I'm saying, hey, we may need to adjust the way that you've been doing things, breeding on your farm forever and ever. It's going to help you, but it's weird and it's different. So don't be worried. I'm here to help. One of the things I found helps a lot with these kind of things and figuring out that level of who you're talking to is you can say things like, some of you may have done this or heard this already, but. And the reason you use phrasing like that is because the people who have heard of it are going to be like, yeah, that confirms what I have done in the past. But if it's new to somebody else, they're not going to feel like you're talking over their heads. So stuff like that is super important. Number 3, make it interesting. Hopefully you have found some of this talk interesting. I really liked using cartoons. This was a fun way for me to interact. These are some cartoons I made up that are on the ICBF website that explain how the TB test works every year. Because those are the things that I heard lots of complaints about is why are we TB testing every year? All of this was done before AI, so it was just me and Photoshop and some really rusty skills. But it got done and it worked out. And farmers said that it helped them understand why and what was going on. So now with AI, these could probably be even better. When you're making your headlines for your popular press or you're doing presentations, try not to make them too sensational. Make them interesting, but don't be too sensational because you never want to overpromise and underdeliver. That is an immediate trust breaker. And the thing that you're trying to do is gain trust. That's the number one thing. If people trust you, they're more likely to engage with you. Lastly, for number 3, if you're going to use a lot of technical images, make sure you use some common language. Make it so that the people who are super interested in it can dig deeper, links for technical information. So if you've got that farmer that has the ADHD superpower of hyperfocus and they want to dive down that rabbit hole, have that information available, but use the common language on these technical images so that anyone can start there and then get more into it. Number 4, make it accessible and open. So these you're probably going to notice some of these kind of layer on top of each other. When I'm talking about accessibility, physically accessible, popular press, websites, farmers go to those. People go to those. There's not a ton of farmers that read peer-reviewed journals. Some of them do, and that's amazing, and that's fantastic. But a lot of them will rely on popular press, trade magazines, those types of things. So make what you're trying to work with them on available in those, and preferably free. As I mentioned before, use common language, so you're making it accessible to everyone with your language. So I would use no back passage versus atrasia ani, because farmers wouldn't necessarily recognize atrasia ani unless they had experienced it before and heard their vet talk about it. Also, think about multi-generational. My reference points, because I'm older now, are way different than the student reference points. And so you have to make it relatable to both current and future farmers. That's really important. Future farmers are going to be the ones that may adopt a little bit quicker. They may be able to talk their parents or uncles or aunts or whoever is running the farm into using your system because they trust you and they want to adopt that. Openness. We are not airing dirty laundry. Again, back to that shame. We're searching for answers to help everyone. Please, no matter what you do, be upfront about timelines and issues. This is a very slow burn. So it's not like you're going to be able to get through this all with everybody very quick. You cannot promise someone, I'm going to have that cause of mutation for you in 2 weeks. It does not work that way. Some of this is still lingering for years, and it's very disappointing, but we're getting better and faster at this. But be very upfront about this. Hey, I'm collecting this. Stuff may not come from it right away, but we're doing our best. If we prepare everyone for those changes, it's not as scary when it comes. If we prepare people for that kind of disappointment, I guess, it's not so scary. And then also talk to them about changes that might have to go on in their breeding programs and why we may change our mind about things. So I want to use a quick Example of the Angus Association, when they first found out about things like developmental duplication, they basically were like, do not breed anything that's a carrier. Don't breed anything that's a carrier together. And that ended up narrowing the gene pool. So now they've taken on this breed smarter, very much so same thing, where they are just choosing who they breed to. Last thing for this one, open means listening to. Now I'm going to go really fast through the next ones because we're almost 45 minutes in. So be prepared to back up your info. Failing to prepare is preparing to fail. If a farmer or a person is resistant, take the time to listen to their concerns and address those concerns. You can't win everybody over, but you can leave them with a good impression of you. So don't fight. You're just going to make everybody mad. But if you listen, you can try and address those concerns and then try and follow up with something along the lines of, does that help or does that make sense? People don't ask questions because they are worried about looking stupid. There are no stupid questions, but everybody worries about that. And so if you can follow up with what they're asking with a does that help or does that make sense, it helps establish a connection and it encourages them to follow up with any questions. And it shows that you're willing to listen. So that's super duper important. And you can always say, let me gather some information for you. You don't have to know everything. And if you find out you're wrong, be sure to tell them, because if they find out you're wrong and you don't say anything, you're going to look really silly. And again, loss of trust. If you find the jury's still out on the science, mention that too. Like, science is ever changing and evolving. It's super important to kind of focus on that. Number 6, don't be afraid to say, I don't know. This is probably one of the most important things. As I mentioned, nobody knows everything about anything. Maybe you know an expert that can help. Grab a phone number, email address, Instagram, whatever you can from a farmer. I don't know, but I want to help. Why did my genetic defect stuff take off? Because farmers remember that funny American lady who wanted to talk to them about their silly looking animals or sad animals, and I would actually get back to them. Word of mouth travels. Lastly, not everybody learns the same way, so target your audience. Knowing your audience will help you pick your battles. As I mentioned, you can't win everybody over, and that's okay. Sometimes folks make decisions on feelings, not facts, like this little calf here who's super cute. You can't change that. So don't force the point and end up risking looking like a jerk. That's not your battle to fight. So lastly, where do we go from here? I want to update and reissue the genetic defect booklet because I found that's been referenced a whole lot and people find it helpful. My feelings are AI companies are global, so genetic defect recording should be as well. If we're sending semen all over the world, we should have this available to everybody in the world. I'd love to develop a system for the USA. That's not in my remit right now, but the hope is the next time my remit comes up, I can add this to it. And I want to partner with other countries to develop a way of sharing this information, maybe come up with a consortium. So with that, very quickly, I'm happy to take any questions you might have, and I thank you all for taking the time to listen, and I hope that you have a wonderful weekend.
Thank you very much, Dr. McClure, for your presentation. I'd now like to open the floor to any questions, either from people here in the room or from those joining us online. We do have a question in the room. Please, Gabby.
Yeah, go ahead.
Hi, Dr. McClure. I'm Gabby. I'm a PhD student at the university working on something similar. And I just had a few questions. First, I guess just to clear up how you were sending this survey to the producers. So you mentioned that you talked to a group of vets at a conference, but I was wondering, how did the producers that maybe didn't hear from a vet first get a hold of the survey? Did you send it like, yeah, how could you explain that a little bit better on how that was?
Yeah, I'm
reported?
terribly sorry. So we actually hosted it on the ICBF website. In the beginning, we didn't have any software that we could use for this. So I literally used the SurveyMonkey software and I created a online link. And so that allowed me to put that link in the popular press. That allowed me to put that link on ICBF. That allowed me to email that link to anyone who wanted it. So I created an online link. I even had it on cards in a tiny URL. So farmers could have access to that. And anytime I went anywhere, I basically would be willing to hand those things out. Online is key, because then you can export that into some sort of CSV file versus having to hand put it in yourself. It has the farmer doing most of the work for you.
Right. And just more on the analysis side, how many conditions or how many reports from the same condition did it take before you went to sample those sequences and analyze the the data there, or did you just do it all at once? When the information came in, you went and sequenced it in those.
So I usually would wait until I had 5 or 6. And if I had 5 and they were related, then I was like, yep, we got something. We should go with this. Even as
Okay.
few as 3, if there was a very clear correlation, heritability type, like I'm seeing this from a single sire, I would be more comfortable. And the same sire across multiple farms. So it's very much so a horses for courses kind of thing. We also had a rash of tailless animals one year. So they had either really, really short, like a corgi tail, so no tail at all. There were ones that had, I have dogs, so I put in like dog terms, the Weimaraner length, which was like 6 inches. And then we categorized those as different. They were all truncated, but they were all different. So we had to wait and see who was related, who wasn't. Are they sending the same ones? And so it's a slow burn. I will say I wasn't comfortable sending stuff off for whole genome sequencing for probably 3 years because the first year I only got 23 records. Second year, I ended up with 50. Following that, it like doubled every year. It was pretty amazing. I was delighted.
Wow. Okay. That's really cool. Thank you. And I know you mentioned that it depended on what you were looking for when you were sequencing versus genotyping. Could you explain why?
Yeah, so if it was something that was already on the chip, we would
Oh,
genotype.
okay.
So we check if it sounded like something that was already known. We would basically use the genotyping as a screen. Hey, does this flag as something we already know? Nope. Okay. Then it goes into we got to do further analysis. And so it'll
Okay.
fall into the whole genome.
Okay. Nice.
And feel
And one
free
more
to
question.
email me anytime. This is my passion project. So
Absolutely.
let me know if there's anything I can do to help.
Yes, for sure. I'm just one more question before I let Colin uh, ask what he was going to. You mentioned the gene editing, where you
Mhm.
can just edit the gene out that's causing the condition. How do you think farmers would perceive this? Do you think they'd be okay with that, or like consumers even too?
That is an excellent question. And that's why I say kind of it's difficult. So we actually tried doing that in the United States. UC Davis created polled Holstein animals and they gene edited them for polled. And you don't hear anything about that anymore because there wasn't great uptake. Now, if this is something that is a extremely expensive fatal fetal abnormality, maybe they would be more open to this. Maybe if it is something that could have a strong economic impact, because I find producers will vote with their wallets, but whether or not consumers are comfortable with it is going to be the drive. So the
Right.
way I kind of explain it is the option is there, but whether or
Okay.
not we're going to actually get uptake in it is very difficult. I'm sorry, that's not a great answer, but that's where
No,
we're
no,
at.
that's good. No, I get it. Yeah. Thank you very much for this talk. It was really great, and I'll probably reach out to you if that's okay. Thank you.
Wonderful. Please do.
Colin.
Colin, what's up?
Hello, Jen. Can you hear me there?
Yeah, I can hear you.
Perfect. Thank you very much, Jen. Fantastic talk as expected. I did have a couple of questions specifically, and they're probably questions that I already asked you before, but I did want to say during my time at the ICBF, they all spoke very highly of you, so they really appreciated all the work that you did while you were there. And as you kind of described, and from talking to them, it was like a one woman mission kind of setting it up. So my first kind of question is now back in North America, do you think it is as it kind of like Ireland's obviously a much smaller place and it's easier to get around? Like, do you think it will take kind of just one person taking on that kind of work to really just try and drive it home? Because I know you started with 23 records or something, you said the first year up to 50. Like, how do you keep the passion going? How do you keep the kind of industry that's kind of funding your work essentially invested in keep on kind of driving this forward while you get the uptake to eventually come back?
So it's a great question. And I actually reviewed this whole talk with my technician before I gave it because she's not familiar at all. And I was like, girl, you ready to take this on? And she was like, yeah, let's do it. So it will no longer be a one-woman show. It will be a two-woman show. But I think, as you mentioned, with the United States, it's much, much larger. There's a lot more cows. But my thought is to leverage the AI companies a little bit more. Here in Madison, we have most of the AI companies that are in North America headquartered here. And so if I can get buy-in from them, because it's going to be a return to them, this is going to be a service essentially to them. If I can get them to help me with this, whether that is with collecting the samples, having them come into them, storing them, and then I come get them after a while, having things as an online format where I can check it periodically. One thing I love is I am learning to code much better with the help of AI. So being able to set up something that will automatically send me a report every week so I can flag things and say, oh, okay, how now I have this many. I feel like it's going to need more than one person to run it in the United States, but I feel like that's where I can leverage the AI companies. I also know that CDCB has been interested in this in the past, and also some of the universities might be interested in collaborating in this, particularly some of the genetics departments may be super interested in this. I worry about getting industry too far involved only because of my comment about patenting the genes. I want this to be free. I want these genes to be available for everybody to use, to put on any SNP chip out there, because I'm a federal worker. I'm here to help the farmers. I'm not here to make somebody else money. So it is, and that's how I felt at ICBF as well. I was working for the farmers. I'm not working for a specific company. Does that make sense?
Yeah, absolutely perfect. And if I could ask one more quick question, Julia, you mentioned the kind of, which I completely agree with, the importance of kind of getting the vets on your side as well as the kind of the journalists. For people that don't know, the Irish Farmers Journal is the biggest kind of journalist for agricultural information. And did you have any difficulties getting buy-in from them? Because during my trip, I heard that sometimes vets, they don't want to report information because it might lead to a bad relationship that they have with a producer, for example, and they don't want to worry about that stuff, so they'll leave it to the producer if they want to report it. Like, did you run into any issues like that, or how did you kind of manage those kind of situations?
I'm going to be honest. I made myself open and out there so much that I didn't have any issues with that. I didn't have any vets come back to me and be like, oh, yeah, we saw this, but they didn't want to report it. And I will say probably 85% of the reports that I got were directly farmer, probably even more, probably closer to 90% were directly farmer. And it's again because this daffy American woman was running around looking for funny looking calves. And Jesus, she's loud, but she's great crack. And so, like, it worked out really well. And just, I never had that issue, but I can see how that could be a problem. So, for that, you can't put their job in jeopardy, but you can encourage them to say, hey, why don't you have a chat? Why don't you have a chat with the person, see what comes up to it, encourage the vet to take a sample. And if you do something like that and the vet is comfortable reporting it, it doesn't have to go directly like, no, I don't want to say that either. You do want the farmer's involvement because then it's too sneaky. And you don't want sneaky. Sneaky is very bad. But if the vet at least takes a sample and holds on to it, maybe they could talk them into it, or maybe you can talk them into it. So it's not lost. It's not buried. But yeah, good luck. That's why I had that primer at the end. This is how you get it.
No, it makes sense. It's good. And they might be just that point of higher education or like giving the education because they have that one-on-one time with the producers and stuff like that. So no, that's perfect. Thank you very much, Jen. That was a great talk. I appreciate it.
Thanks so much. Thank you very much for having me.
We do have one last question here in the room.
Yes, Jennifer, here's Flavio Schenkel. Thanks for the presentation. It was excellent. And I like to pick your brain in something that I'm thinking in the last couple of years, is that how to manage all these very large number of defects in unfavorable haplotypes. that every year we are discovering more, right? So it's getting more and more difficult to include that information in the selection process, but also in the mating systems.
Mhm.
You mentioned to use AI to help this puzzle because it's a puzzle, right? Or you are thinking more in a more traditional way, like create an index of defects, where you would weight the defects based on the impact, economic impact, but also the biological importance of the defect in terms of the animals. So how we are dealing with this? What is your thoughts? How we can do this in an easier way that we are dealing with nowadays, right? That is really, really difficult to weight the
Yeah.
different defects.
So this can kind of lead back to that gene editing bit. So in a perfect world, there wouldn't be pushback, and you would edit some of these out of the AI population. Because then, even if you breed them to carriers, you know you're only going to get a 1 in 4 chance of getting a carrier out of that mating. So then you end up slowly pulling it out of the population. Since that's not really an option at this point, mostly what we have focused on is on the selective breeding, identifying your carriers, and then having as many AI animals out there as you can. And possibly if you know that you have a cow. So we'll take a dairy cow. This is an easier one for me. So you have a dairy herd and you have one cow and she happens to carry Holstein haplotype 1 through 4. She's got all of them. She may be a good producer, but she's not a good economic bet because you're not going to be able to get her pregnant. You're going to have a much harder chance of getting her pregnant if you can't find a sire that is negative for one of those. So maybe instead of becoming a dairy cow, heifer replacement cow, she becomes a beef producing cow and you breed her with a different breed. You would end up having to possibly bring in crossbred animals to do this effectively. It depends on what the farmer's primary objective is. If their objective is this cow that produces a whole lot of milk and getting milk out of her and doing that, then you breed her to beef. If his objective is to create replacements from her, he may have to look at a different dairy breed as a crossbred, or take that chance and just educate the daylights out of them, saying, you can do this, but you have a 50% chance of losing that calf based on X, Y, and Z. So if they end up breeding the carrier to carrier, 1 in 4 chances you end up producing an animal that's not a carrier anymore. So hope is not lost. It's just creating the best strategy. Now, in terms of a genetic index, that's where you're starting to talk about, okay, what is her breedback value versus not, and putting that in almost as a reproductive element of it. And that could play a part in it as well. But for the most part, I kind of look at it as like a dating service. So they are picking the best sires for the best outcome based on what that farmer wants to do. Does that help?
Yeah, no, I think it's a great discussion, this, and I think it will be even more important in the future. But what I refer with the AI was not artificial intelligence, artificial insemination was artificial intelligence.
Oh, yeah.
To try to figure out how we can best combine all the information that's available for sake of selection and use of the animals. But
yeah, so for
artificial
intelligence, what I see is using this for machine learning and being able to increase your imputation speed so that you have a better handle on what genotypes are out there. Because some people are moving to skim sequencing, which is a ton of data, and that's going to require a ton of processing. So we need to actually utilize AI to speed up some of these processes. So that's kind of where I was thinking of using AI, was as a tool to increase processes, less of a tool to develop that index. But it's a possibility too.
Yeah. Okay, I just like, it's not a question anymore, but I like to say, I maybe on behalf of the group here in Guelph that are working on this, I think we are really open for collaboration, and we will be, I think, happy to collaborate if you are open to, because
Definitely.
Gabi's project is really to start putting together something to for a national level in Canada, or recording all the pipelines and how to analyze the information that would come from the farmers. So, and you have all this experience already, so it will be very, very useful for us if you are able to, we're open to collaborate.
I would be delighted. Thank you.
Okay. Yeah, that's good. Thank you so much.
It has been a great discussion. Thank you, everyone, for coming. Thank you again, Dr. McClure, for presenting for us. The recording will be posted on YouTube, and we'll share more details about that with you later. And that's all for today. Thank you very much. See you. Have a nice
Thank
weekend.
you.
Thank you so much. Bye-bye.
Bye.